lot of water. And we to drip when we give, because the drip system is also allowed in case of organic farming, because here under restricted conditions we are using this drip pipes, because after completion of the crop we should not left over the drip lines. It should be properly disposed of from the field. So that is the main important point to be noted. And according to the growth stage, the requirement of water for banana is after planting of suckers, it is 5 to 6 liters, and early growth stage it is 8 to 10 liters, and grand growth stage that is 12 to 14 liters per plant. And at bud initiation stage it requires more water, that is 18 to 22. After shooting to bunch development it requires 25 liters and above. So depending upon the age and season of the plants, we have to irrigate the crop. Coming to the nutrient management, banana is a voracious feeder of nutrients and respond well to the applied nutrients. Here we are using different organic substrates, biofertilizers, and restricted use of rock phosphate. Under restricted conditions, we have to use rock phosphate and sulfate of potash. Sprays can be done under organic farming. These are the potential of in India. We have a lot of potential to use as organic. For so many sources are there, organic sources like livestock and human waste, and but we should not use human excreta, and byproducts like slaughterhouses and crop residues, tree waste and aquatic weeds, green manure crops, and urban and rural waste, and agro industries byproducts like oil cakes can be used, and marine waste, fish meal and seaweeds, and tank silt. These are all the potential organic sources. A lot of scope is for the application of these organic sources in India. So there is not availability is, and wider scope is there for the organic production of banana. And coming to the nutrient management schedule, which was developed at Hort and at the ASRP fruits. First of all, we have to go for at the time of planting, we have to add FIM 10 kg, neem cake 1.25 kg, arbuscular mycorrhizal fungi 25 grams, azospirillum 50 grams, PSB phosphorus solubilizing bacteria 50 grams, Trichoderma harzianum 25 grams. All these has to be mixed, and we could add at the time of planting itself. This is per plant per pit. And next, followed by the triple green manuring. We have to grow the green manure crops 3 times. Started with along with the plantation, we have to raise the green manure, that is sun hemp, and incorporate it 45 days after sowing of the sun hemp. Later on 10 days we will give the gap, and again we will sow the cowpea seeds, and after 30 days of crop growth of cowpea, we have to incorporate, again we have to incorporate in the field. And the next third time, at 10 days interval, again we have to sow cowpea, so incorporated the 30 days after sowing. So in this way, we have to grow the triple manuring for 3 times. So this we should complete green manuring within 150 days. That is, within 5 months we have to complete the green manuring. Otherwise, there is a detrimental effect to the crop growth, because in that within 5 months after planting, there is a gap. And also there will not be any shade effect on the green manure crop also. It not only this green manure crop provides nitrogen fixation, and also it is smother the weeds. It will not allow the growth of the weeds. And at third month after planting, we have to add vermicompost at the rate 5 kg per plant. At 5th month after planting, we have to add wood ash 1.75 kg per plant. If it is not available, in some places it is burnt rice husk is available, which is available at the hotels, and this can also be used instead of wood ash where it is not available, at the rate 3.5 kg per plant. All these come together. When we calculated the NPK, it comes to N 200 grams, P 50 grams, and K 200 grams. So that is the recommendation actually we are giving for the inorganic fertilization. Same recommendation when we can supply through this by this allowing this nutrient management schedule for banana, and by adopting these practices. And the same brand name was developed under ICR ASRP fruits. And this is the bunch that was developed, which is on par with the bunch that was developed with the inorganic. but it is less than organic, the bunch that is formed in under organic is less than the inorganic, but it is on par with the inorganic treatments. And during in earlier days also, that is during 2005 itself, we have at our station we have developed a sustainable organic production system, and in granite itself, that is for both plant and return crop. The plant crop with this experiment is conducted only with the organic residues, the organic products application of either farmyard manure along with poultry manure or vermicompost and farmyard manure per crop cycle. Each crop cycle is used for the raising the plant crop. Same can be applied in split doses for the return crop also at monthly intervals for 4 months, allowing the one follower sector. And also by adding the biofertilizers along with this organic residues, also we could use organic manures plus biofertilizers. One more technology was developed, that is application of either poultry manure or farmyard manure along with azospirillum, that is and AMF, that is arbuscular mycorrhizal fungi, was found promising for the production of organic banana cultivar granite. And also not only in granite, in the local cultivar that is proven, that is Karpura Chakarikeli, best organic farming protocol with respect to the yield, quality, disease incidence, soil health and cost economics in banana Karpura Chakarikeli was standardized for alluvial soils of Godavari zone. That is application of poultry manure 8 kg per plant in 4 split doses, that is 2 kg each at monthly intervals, along with azospirillum 50 grams per plant, arbuscular mycorrhizal fungi 50 grams per plant, recorded highest yield and quality. These are some of the organic production technologies that are developed for banana at our station. That is in Andhra Pradesh. Other than Andhra Pradesh, that is at NRC Banana Trichy, application of 5 kg poultry manure, 1 kg groundnut cake, 3 kg rural compost, 3.75 kg wood ash per plant was found promising for the production of organic banana, that is cultivar granite. And at KU Thrissur, the application of 28 kg FIM or poultry manure 14 kg per plant or vermicompost 10 kg per plant along with the wood ash 4 kg per plant in 2 splits, that is one at basal dose and one at 3 months after planting, and also 0.5 kg lime as basal in situ green manuring was found promising for the organic production of Nendravi. So these are some of the organic production technologies for nutrient managements were developed in various states. And coming to the weed management, raising green manure crop in the interspaces and incorporating in situ 2 to 3 times for weed management and nitrogen fixation is suppresses the weeds. And also, if at all it required, after 5 months, we can go for the 1 or 2 manual weedings also. We should not disturb the soil after bunch emergence. And mulching is an effective practice for controlling of weeds. Organic mulches like banana trash, sugarcane trash, whatever, and paddy straw, whatever may be the organic farm residues are available, they can be used at the rate of 15 tons per hectare. And also we can use the live mulches like growing cowpeas and hemp, etc. We can add the, we can smother the weeds. And also we can, under restricted conditions, we have to use the plastic mulches. But after completion of the crop, after removal of the crop, we have to remove this and dispose in the safe manner. That is the main point to be recorded. And next, coming to the bunch management practices, there are after so many, we are mostly the organic products are exporting to the other countries, or they are fetching more price. So it should be taken more care than the normal practices. So we have to go for the denaveling, that is removal of the male bud should immediately after the bunch last hand, opening of the last hand. It not only by removing the male bud, we can decrease the maturity period from 7 to 10 days in bunch. And also we have to go for the peduncle wrapping. During summer months, what happens is whatever the peduncle is exposed to the high temperatures or scorching temperatures during summer, then at that portion the tissue is damaged and weakens, and then ultimately the bunch may be dropped from the plant. So we have to go for the peduncle wrapping, covering the peduncle with boot leaf to prevent the sunburning or scorching. And then next comes to the bunch covering with dried banana leaves. It is a age-old practices that is followed in all the traditional areas. And especially in case of red banana, if we use these bunch covers or these leaves, the color of the red color is more predominant compared to the exposed ones. So that is the advantage. And we can also go for the polyethylene, polypropylene bunch covers, bunch sleeves, but we should not use the polycarbonate in case of plastic uses low. We need not use this polycarbonate material, but we can use under restricted conditions polyethylene and polypropylene bunch covers. But we should not use the pesticide impregnated poly covers. And coming to the bell injection with azadirachtin 1%, that is 10,000 ppm azadirachtin that can be used under restricted conditions. 5 ml of 1% azadirachtin can be added to the diluted to 1 liter of water. From that solution, we have to take 2 ml of that solution and injected into the bud. When the correct stage of the bud injection is it should be straight, that is, it is just emerging from the from the plant, and it should be straight. It should not bend. If it is bent, the whatever the material we are injecting will not go into the plant. So it should be straight enough. This is upright in position, and we have to give the injection from the topmost 1/4 portion of the bud at 30 degrees angle. Then only that that whatever the neem oil, that is azadirachtin neem oil, we can use. For it is used against sucking pest, especially thrips. We can see the what are the thrips effect damage, and this is without the azadirachtin bunch injection, how it looks in Katpur Chakrakeli. And next is the removal of the floral remnants. This is also an important operation, wherein in some varieties like Amrutapani or silk group, the floral remnants were persistent for a longer period in the bunch. So they will act as a host for the thrips as well as for the diseases. Diseases like cigar and rot. During the rainy season, if the water is present at that floral ends, then it will cause cigar and rot, especially in Telachakrakeli, Kovurbonta, and also in granite. It is this disease was observed. So we should remove the floral remnants. And in summer also, this at the floral remnant portions, the thrips may be hidden, and they will cause the damage to the they will spoil the appearance of the, though the quality is not reduced, but the appearance of the fruit is reduced. So hence we have to remove the floral remnants in both the seasons. And next important operation is propping with either bamboo or casuarina. We can go for propping, especially in the coastal districts, where coastal areas where the winds or gales are high in case of in summer, and summer also cyclones and cyclone prone areas also. In banana, it is an important operation to be done. And next, coming to the desuckering at regular, we have to do for desuckering at regular intervals, and because from second month onwards, depending upon the variety, the suckers may come, and we have to remove those suckers at regular 20 days interval, so that they will not harbor any pests and diseases. And the earthing up is also essential. And next, under restricted condition, we can use a sulfate of potash under organic farming at the rate to 20 grams per liter on bunches 2 times within 15 days interval, so that the bunch will be, because the bunch requires lot of potash, so it needs extra potash. So we can give the sulfate of potash to the bunch sprays. And next, coming to the one of the component in organic farming is the inclusion of diverse crops. So that can be done with the intercropping. So intercropping of banana with especially tuber crops is beneficial, because this colocasia and elephant foot yam are the tuber crops, being these are well suited under the banana's agro ecosystem, and they will respond to the organic farming also. along with the banana. So these two intercrops are beneficial in banana as an intercrops. And coming to the pest management, the major problem in banana cultivation is pseudostem weevil, rhizome weevil, and especially this is not occur in Andhra Pradesh. Nowadays it is also coming, but in neglected gardens it is observed. Coming to the pseudostem weevil management and the clean cultivation, that is removal of the dry leaves and removal of severely infested plants along with the rhizome, and we have to destroy them and spray neem oil at 5% and plus garlic to the leaf axils and pseudostem during your 3rd and 5th month after planting. Application of Bavaria bassiana at the rate 20 gram per liter from 5th month after planting, and leaf axil application of entomopathogenic nematodes at the rate 4 cadaver per plant from 5th month after planting is also effective in controlling the pseudostem weevil. And so spread pseudostem traps, that is swabbed with Bavaria bassiana at the rate 20 grams per trap from 5th month after planting also can control the pseudostem weevil. And coming to the rhizome weevil, which is present in the rhizome, and it is tunnels the rhizome, and thereby the plant is weakened and sometimes it is fallen down also. So this for the management of rhizome weevil, we have to go healthy planting. At the time of selection of the planting material itself, we have to see that whether rhizome weevil is there or not, because it is visually seen by seeing the tunnels in the rhizome portion after removal of the suckers. So here also the pairing and the dipping of suckers in cow dung and slurry and ash is essential, and field sanitation is important aspect. The pseudostem traps of 50 centimeter length can be used in the evening, and morning we have to collect the weevils daily and we have to destroy them. Otherwise, that will spread into the other portions. So, collection of the weevils is an important aspect. And sucker treatment with Pseudomonas fluorescens, that is 20 gram per liter, and entomopathogenic nematode infected cadavers at the rate 4 cadaver per plant at the time of planting is can be applied, and application of same at 2nd and 5th month after planting also. We can use cosmolures and application and can apply neem cake at the rate 1 kg per pit and drench the soil with this entomopathogens, that is Bavaria bassiana, Metarhizium anasopliae and entomopathogenic nematodes, so that we can control the rhizome weevil with the with organic farming. And next minor pests or aphids, this can also be controlled by biocontrol agent using biocontrol agent Verticillium lacani applications. And coming to the nematodes are the major problems nowadays because of the adoption of the drip irrigation also, because these nematodes live in for their existence they need water. So under drip irrigation there is a continuous film of water, so they can survive in the under drip irrigation system. So the problem is going on increasing. So for the management of nematodes, we have to first, before we have to planting, we have, if the field is infected with nematodes, we have to early before planting itself we have to grow marigold or going marigold at the basins, or intercropping the banana with sun hemp or marigold. It can reduce the nematode population, and pairing and hot water treatment is also very effective in controlling the nematodes. And application of neem cake at the rate 1 kg per plant at the time of planting, and also basal application of nematopathogenic fungus, that is Bacillomyces liliensis at the rate 25 gram per plant. Or we can use the consortia of both Pseudomonas Bacillomyces and also the Trichoderma harzianum. We can separately multiply in the FIM, and later we can mix and apply it to the plant. For the nematode, can we control the nematode effectively? And next, coming to the disease management, among the diseases, Sigatoga or New-Mesa leaf spot is an important disease that is being observed in banana. Some of the cultural practices, mostly by cultural practices, we can reduce the inoculum load by proper sanitization and by timely weed management and proper drainage, clean cultivation, timely desuckering, and by adopting optimum spacing. Especially the Cavendish banana such as grandin is highly susceptible to disease. If grandin has to be selected, then it's the grand growth stage or the shooting stage. Shooting means it is when the bunch appears. Bunch emergence stage, that should not coincide with the rainy season to avoid disease. These are the important cultural aspects to be reminded. And among the biocontrol Management, biocontrol agents like Pseudomonas at the rate 20 grams per liter or Bacillus subtilis 5 grams per liter can be used. And under restricted conditions, we can spray initially, the initial stage of the disease, we can spray 1% border mixture or petroleum-based mineral oil 1% in the under restricted conditions only. And next, coming to the rhizome rot, this is the main problem in Telechakkarkeli, Granite, and Kovurbonta, which are mostly susceptible to this Erwinia rot. And avoid planting these varieties during summer months, that is from February to July. So, growing resistant varieties like we can go for the Karpura Chakkarkeli and use of healthy and disease-free plant material, because this is a bacteria. It is coming through the plant material and the soil adherent to the roots or suckers. It will come into the newly planted field. So we should take care with the. We should not select the plants from the diseased mother blocks. And growing wet paddy or sugarcane preceding banana will ensure against the incidence of rhizome rot in banana plantations. Badly affected clumps should be destroyed after uprooting the palm. Here we can see that this is also called as toppling disease, because at the rhizome region the bacterial rot will occur. So by touching just the plant, it will topple down. So we have to remove such type of damaged plants, otherwise they will spread to the new plants also. And as at the restricted condition, we can use COC and for sucker treatment only, and drenching with 50 grams of Pseudomonas fluorescens per liter per plant, or 2 to 2 drench at monthly interval. It can suppress the inoculum. And also application of Pseudomonas fluorescens at Trichoderma multiplied in FIM at fortnight and applied at fortnightly intervals can check the severity of the disease. The disease spread can be reduced through the by adopting the drip irrigation, because the bacteria can spread through the irrigation water also when we give the flood irrigation. So we have to go for the drip irrigation and ensure optimum water during the summer. These are some of the management practices to be done for the control of the rhizome rot disease. And coming to the other important disease, that is Panama wilt. First of all, we have to go for the resistant varieties, that is the fire hybrids, and also Yangambi KM5, Pisancillium, Williams, like GCTCV 119, GCTCV 215. The cultivars like dwarf Cavendish, Granite, Karpura Chakkarkeli, Robusta, Red Banana, and KBS 8 NRCB1 are tolerant to the disease. So we have to go for the tolerant or resistant varieties instead of susceptible varieties. Rastali group, that is especially Amrutapani cultivar, is highly susceptible to disease. However, the cultivar Marthaman is moderately tolerant to disease. So we can go for this variety, which is similar to the Amrutapani. This is also called as Bengal Amrutapani, Marthaman. And select the plant diseases free materials. Select the plant disease-free materials and avoid ill-drained soils, and badly affected clumps should be destroyed after uprooting the clump. Apply 500 grams lime per pit, or apply 1 kg neem cake per pit at the time of planting, or apply AMF, Pseudomonas, Trichoderma harzianum, or PGPR mix at the time of planting itself. Because this is a soil-borne diseases, we have to apply more and more biocontrol agents to the soil region itself. And we can do sucker treatment with Pseudomonas at the rate 20 grams per liter. And coming to the important viral diseases that are affecting the banana, that is banana bract mosaic virus, which the symptoms which we can be observed on the bract itself, or otherwise we can see on the spindle shaped spots on the stem itself, and the banana bunchy top virus, and also the cucumber mosaic virus and banana streak virus. These are the 4 major diseases that were observed in the banana cultivation. Use for the whole for the management of whole viruses, we can use the healthy disease-free planting material. We should not take the for as a marker for banana bract mosaic virus, we can observe while taking the suckers itself from the whenever the bunch is there, because on the bract itself, that is on the male bud, we can observe the disease symptom. Then we should avoid those fields for taking the suckers. And avoid growing cucurbitaceous crops as an intercrops, because these cucurbitaceous crops act as a host for the CMV mosaic virus. And eradicate infected plants as and when observed, and clean cultivation and weed control is essential, because most of these are transmitted by the aphids, and which these weeds can act as an alternate host for these aphids, and also mealybug. Banana stick virus is transmitted by mealybug. So alternate posts will be destroyed. And use neem-based insecticide to control the vector. And for aphid control, we can use a biocontrol agent that is Verticillium lacani. So with this, all the management practices, we can control the viral diseases in organic farming. And this is in a nutshell, the pest and disease management, that is select pest and pathogen free. For all the disease and pest management, just I am giving the conclusion that select best pest and pathogen free planting material. Pairing is essential, and immersion of suspension containing entomopathogen fungus, that is metarhizium, Anaceplea, and Tagetes erecta leaf extract. Application of bioagents like Pseudomonas fluorescens, Trichoderma hartianum. We have to use them at frequent intervals, and adjusting planting time. We should not coincide with the rainy season. The bunch emergence should not coincide with the rainy season or summer season, and we should not plant during the summer season also. The susceptible varieties. And spray neem kernel extract. Neem oil may be used under restricted scale, and maintain optimum plant population, drain out excess water, and maintain sanitization in the field. And whatever the dry and dried leaves or diseased leaves, we have to cut and burn them. And prune suckers below the ground level to prevent the aphid infestation, and apply Pseudomonas fluorescens or Trichoderma viridi or Metarhizium anaceplea multiplied on neem cake. And on a restricted basis, we can use border mixture or copper oxychloride. And coming to the harvest, the correct stage of harvest is essential. For long distance transport, we have to go for 75% to 80% maturity. And for short distance transport, we have to go for the 90 days, 90% maturity stage. So depending on the need, we can harvest the crop. And also the careful harvest and dehandling without the bruises, because in banana most of the bunches are harvested and transported as a bunch itself. But nowadays, by removing the hands, that is dehandling is adopted to getting the best prices, and also do easy handling without the much damage. So coming to the post harvest, wash the banana hands in potassium alum or extracts from the lemon, orange pips, or extract of Thymus vulgaris. And later we can wet the cut end with vinegar to prevent the crown rot. Last time we have seen that under restricted chemicals, this vinegar is there. We can use it only under under the supervision of the certifying agency only. But so that in order to prevent the crown rot, which is appearing in the as a post-harvest disease, and to delay ripening kaolate, and for ripening ethylene gas can be permitted. So in packaging also, the material, whatever the material that is used for packaging should be biodegradable and reusable, and should not contaminate the food. These are the standards which we have to maintain to get the best organic product in banana. And next, first we have observed what are the production aspects of organic banana. Now it is important to know about just a brief about the certification process of banana, which it has to be for what are the standards we need to get the certification process. Certification, what are the benefits of certification or aim of organic certification is, it provides the identifiable label in which the product should contain a logo or symbol, so that we can identify, the consumer can identify that it is exactly the organic product, and to give an assurance to the consumer that the product is truly organic, because it is gone through the different certification procedures. So it is a reliable one. And the steps in certification process, it should be fulfill the requirements of first conversion period from inorganic to organic farming, and develop a comprehensive operation plan for the farm, including ecosystem management, convert the farm completely or comply with the regulations in the case of split and parallel operations, that is organic or conventional, and yearly on-farm follow-up inspections made by the certifier, and continuously update the chosen certifier on major changes, and keep track on inspection certificate issued by the certifier. So we have to maintain whatever the farmers are doing organic farming, they should maintain the proper farm records for evidence as it is produced under organic system only. And an NPOP organic system, which is implemented by EPIDA under Ministry of Agriculture, Ministry of Commerce and Industries. So they have appointed some accreditation agencies for organic certification. These are EPEDA, Tea Board, Coffee Board, Spices Board, Coconut Development Board, Directorate of Cashew and Coconut. And these accreditation agencies identify the certification agencies. In turn, they will identify the certification agencies. They certify the organic product and give the label India Organic. And these are some of the certification agencies in India. These are also operating. There is Indocert in India, Demeter Association USA, Organic Crop Improvement Association USA, IAMCO Switzerland, Ecocert France, Belgium, and Germany, etc. And the markets for organic commodities, most of these markets are European Union and North America and Japan. These are the major markets for organic commodities. And what is the benefit of going for this organic certificate for this banana organic production? Because it expected output is, it is maybe direct or indirect. Through direct effect, we can promote the exports, and the farmer can get high returns to the organic farming through the organic farming. And indirect is production of residue-free healthy banana and reduction of environmental pollution and conservation of biodiversity. So this is all about the production of banana under organic farming. It includes the certification as well, the process of certification. Thank you.
Thank you, madam.
Thank you, sir.
If any questions are there, please ask. Students, I request students, if you have any doubts or questions, please ask. I think no one is posted, madam.
Okay.
Doubts?
Is there any profit? Some chat box is there, no? In that.
Yes, sir.
Is there any profit under organic farming? First, in initial few years, that is 2 years of conversion period, we don't get any profit. But after that, we can get the yields on par with the inorganic farming, similar to the inorganic farming. So it is profitable after 2 to 3 years after conversion period only.
Madam, one more question.
Okay.
In chat box. Why we prohibited the use of tobacco leaves in organic banana production?
Tobacco leaves in the organic banana production. Some of the, because these all standards were given by the MPOP standards. MPOP standards, they have depicted in each and every plant extract, they have given this plant material we should use, and others were restricted. So accordingly, we have to use under the certification agency guidelines only. Does organic banana last longer? Yes, the shelf life of banana grown under organic farming is much more higher than the conventional chemical farming.
Any more questions? Madam, I think no one is posted. Hello, madam, am I audible? Madam?
Sir.
I think no one is posted, madam. After one more question is.
Okay, sir. I'll see. You are not, sir.
Is there any
Voice is not so clear. Is there any technique to control nematode attack organically in banana? Yes, we have first from the initial stage itself, we have to do some practices like pairing of suckers, then application of this Pacillomyces liliaceus, these biocontrol agents like Pseudomonas at the time of planting itself in the pits. Then only we have to go for the planting of these suckers. Then we can reduce the nematode population. And also we can, if we plantation, if the plot that is we are going to plant in banana, then we have to grow for the marigold as a early crop. After growing marigold, then we can go for the banana so that the population of the nematodes is reduced. So these are the some integrated holistic package of practices we should follow. Not at the with a single practice we can't control the nematodes. From planting to the till the end of the stage, we have to recurrently apply these biocontrol agents at the base of the plant. Is it clear? Okay.
So, any more questions, dear students? Otherwise, we conclude the meeting.
Thank you, sir, for giving me the opportunity to the university officials and Eddie madam. Thank you.
Madam. Okay, madam. Thank you very much, madam. Just wait, madam. Just I want to give one vote of thanks, madam. So on behalf of organizing committee, College of Horticulture, Venkataramanagudam, it is my privilege to extend my heartfelt thanks to Dr. R.Nagalakshmi madam, Senior Scientist, HRS Lab. Madam, you explained the topic organic banana production in very well effectively and well understood by our students also, madam. You covered all aspects from initial soil conditions to harvesting, even certification process of banana, certification agencies and market for organic banana. So all aspects you covered very effectively, madam. I thank once again from our organizing committee. Thank you very much, madam. And you also cleared all doubts raised by our students also. Thank you, madam. Thank you very much. And also convey my sincere thanks to participants, all the students and staff from HRS, Kovuru Research Station and other teaching staff and scientists. Thank you one and all for participating in this program. Thank you very much.
Thank you, sir.